Hello, everybody.
Let's
We'll
wait
wait
a few
1
minutes.
minute so everybody can enter in the meeting.
And we are ready.
Can we start?
Yep.
So, hello, everybody. Welcome to EMN Live Webinars. Today we'll have the section 28 with Dr. Mónica Cala. I'm Carolina. I'm from Brazil. I'm part of EMN committee members. And with me is Juan José from Mexico. You can pass, Juan, please. So if you want to connect with us to be updated about the next webinar or news about EMN, please join us in the social media. Here we have the LinkedIn, X, and other medias, or you can send us an email. And just for information, we will have the first EMN symposium come soon. So join us in the social medias to be updated about this first event. It will be completely online, virtual. And soon we will have the Metabolomics 2026 in Buenos Aires in just 11 days. So if you didn't do your inscription, please join us in this events. And if you already did it, I will see you there. So it takes only 11 days left until Metabolomics 2026. And now the webinar will receive Dr. Mónica Cala talking about using metabolomics to improve industrial food process evidence from a cough case study. So please, Juan.
So today I have the pleasure of introducing Dr. Mónica Cala, a leading researcher in metabolomics and a pioneer also of metabolomics infrastructure in Latin America. Dr. Cala is the head of MetCore, the first metabolomics core facility in Colombia, established in 2019 at the University of Los Andes. She is also a founding member of the Latin American Metabolomic Profiling Society, or in short, LAMPS, playing a key role in promoting metabolomics research and collaboration across the region. With more than 15 years of experience in bioanalytical sciences, she has expertise in developing and validating analytical methods using advanced separation techniques and mass spectrometry. Throughout her career, Dr. Hala has been dedicated not only to advancing metabolomics research, but also to expanding the metabolomics technologies in Latin America. Under her leadership, the MetCore has become a referee center for both targeted and untargeted metabolomics, providing access to the state-of-art platforms, including GCMS, CEMS, and UHPLCMS. Her contributions to the field have received international recognition. In 2023, she received the President's Award from the International Metabolomics Society, becoming the first Latin American scientist to earn this distinction. More recently, she was recognized by Chemical and Engineering News as one of the Latin American trailblazers, highlighting her scientific impact and leadership in the region. Finally, she is also one of the organizers of the Metabolomics Conference 226 in Buenos Aires, Argentina, which, as Carolina said, will take place in just 11 days. This conference is particularly special as it will be the first metabolomics society ever hosted in Latin America, representing an important milestone for the growth of metabolomics across the region. So Today, she will share how metabolomics can be applied to improve industrial food processes, presenting insights from a coffee case study. Without further ado, please take the stage, Dr. Mónica.
Thank you, Juan, for this kind introduction. So let me share my screen. I think it's working.
Yeah, it's working.
Yeah. So good morning, everyone. Thank you again, Juan, for this kind introduction. And thank you, Carolina, also, and all the EMN organizers for the invitation to be part of this webinar. I think it's an amazing opportunity to present some of our work in food metabolomics. For today, I select coffee as a case study to present some application of untargeted approaches in industrial food processing. But before I start my presentation, I would like to take a moment to introduce our institution and also our Metabolomics Center in Colombia. So MetCore is under the Vice Presidency for Research and Creation at Universidad de los Andes, also known as UNIANDES. UNIANDES is one of the best universities in Colombia, always fighting for the first place, and is one of the best also in Latin America. And Uniandes is located in the heart of Bogota, Colombia, which is the capital city of our country. And as Juan mentioned, MetCore was established as a core facility. I think the concept of core facility is quite common around the world, but in Colombia and actually Latin America, I think is quite new. So in Uniandes, we have these 4 facilities for metabolomics, for genomics, microscopy, and also in animal care. And right now, I'm the head of all core facilities, and I also lead in MetCore. So as Juan also mentioned, MetCore is the first metabolomics center in Colombia, one of the first in Latin America. It was established in July 2019 with the main goal to provide access to cutting-edge technology in metabolite analysis. So the main idea was to boost metabolomic research in our country and also in our region. So we have a platform that provides a complete metabolomic solution from sample preparation to sample analysis and also to data processing. We have a mass spectrometry-based metabolomics platform to conduct this analysis. So here we have mass spectrometry instruments coupled to the different separation techniques, gas chromatography, liquid chromatography, and also capillary electrophoresis. Right now we have 3 high-resolution systems, 2 QTOF systems, one coupled to GC and the other one coupled to LC, and we are running our new machine that we are really happy to have. That is the first hybrid system in Colombia and also the first IQS installed in Latin America. And since last year, we are working to increase also our capabilities in metabolite identification. So right now, we offer analysis with level 1 of identification to or around 2000 metabolites. So we want to ensure more confidence analysis and also, as we are a core facility, faster analysis. So what we do in MetCore, we do different things. The first one is to provide specialized services. Also, we conduct research and also we conduct a lot of training activities in metabolomics and lipidomics analysis across the different fields from biomedical, plant, foods, and also microorganisms. So we have different services from the untargeted approach in metabolomics and lipidomics, some metabolic profiling like amino acids, phenolics, compounds, flavonoids that are really important in foods applications. And also we have with these libraries some services for metabolite characterization. And right now we're working in targeted analysis in applications in biofluids. So we are implementing the mega kit with from Metabolomics, and this is in cooperation with Node and also with Professor Yamile Lopez. If I remember well, she conducted a seminar this year during this topic. So one important thing is for us is boosting this metabolomic research in Colombia. So we collaborate with different groups around the country and the region in biomedical application, mainly looking for biomarkers for disease diagnosis, prognosis, and also to understand pathophysiology across different applications, including diseases, cancer. Of course, we're in Colombia, so tropical diseases, Zika, dengue, among others. And we worked last year a lot in exercise physiology and also about microbiota. In Colombia, one of the most biodiverse countries, of course, we work in bioprospection, and here we are using metabolomics to explore um, alternative phytotherapeutics from our biodiversity to treat some diseases, and we are applying metabolomics since from the metabolite extracts to the cell lines and also animal models and also in clinical studies. And the foods, that is the topic that we are going to focus today. We work a lot in food resources, in industrial food processing. In food resources, we work with Agrosavia, which is the national institution for research in agro in Colombia. So we have a lot of projects in food processing and in food production, and in industrial food processing, we have been working with different companies, mainly in cocoa and also in coffee. So we are going to talk more from coffee. So as a short introduction about the application of food metabolomics, I think it's well known that food metabolomics has been applied to all stages of food production, from food resources to industrial food processing and also food intake. And in food resources, metabolomics is used to investigate the metabolic characteristics of foods associated with the environment and also with the genetic factors. In industrial food processing, we use metabolomics to evaluate and monitor the effects of conventional industrial processing techniques. And in food intake, we use this tool to explore the interaction of food systems and also the human health and diseases. So today we're going to focus in this application. And the first thing is to remember what means food processing technology. So food processing is defined as all techniques and methods used in food industry to convert food resources into valuable products for consumers. So we perform these techniques with different objectives, for example, to increase food safety, to change or modify food textures, taste, flavors, colors, and also to provide nutritional balance. So I want to give a few introduction about the different types of food processing techniques. Some of them are categorized as minimal processing methods because consumers always demand for minimal processed food with a fresh taste and also appearance. So technologies that are characterizing this topic is, for example, vacuum storage, with that quite common using to preserve the foods. And we can use metabolomics to identify metabolite markers or key metabolites in the different process, also to optimize process condition, and also to predict shelf life and evaluate and understand the degradation mechanisms that are involved in this food production. So in general, metabolomics enables a deeper understanding during the minimal processing, helping to preserve quality. And I think the most important task is to extend shelf life. We also have processing technologies related with heat and also with treatment, with dry treatments. And it's well known that when food are submitted to thermal processing, it's possible to have adverse effects in the food companies, maybe nutritional losses and also formation of toxic or undesirable compounds. So metabolomics helps to choose the best condition and also the best operational parameters in industry, for example, time and temperature, to ensure also the product consistency and the understanding of difference between the fresh food and also the processed food. And it's the same for drying technologies, where the main focus is to exploring shelf life and preserve quality. So the idea here is to use metabolomics as a guide for optimization of heat and dry conditions to preserve the nutrients and to reduce the undesirable compounds. And I think the goal is the fermentation technology. I think it's one of the most used and is an all natural economical method for processing and preserving foods. It's really useful because this fermentation process enhances flavor, nutritional value, and also digestibility in food products. So here we can use metabolomics to predict quality in this process, to monitor step by step during the fermentation process, to understand better the microbiota, not only the microorganisms involved in the process, but also the metabolites that are produced by these microorganisms, and of course, to optimize this process. And one of the last categories is the chemical and enzymatic treatments, in which metabolomics helps to connect the enzymatic activity, all the metabolite formation and the microbial dynamics for a better control, flavor, and also functionality in foods. So, as I mentioned at the beginning, we have been working on different applications of metabolomics in food science. So today I would like to focus on coffee. I select coffee because it's one of the areas where I have been applying metabolomics the most, especially to study the industrial food processing techniques in different stages. So I think that it's well known that coffee is one of the most consumed beverage in the world, and it's also a product of great economical, social, and cultural importance. And when we talk now about coffee, we observe that coffee has evolved from a basic commodity to an expression of art and craft. This means that coffee transformed the international trade from regular coffee products and also or commercial coffee to specialty coffees. And in coffee, the price is highly related to the quality of the coffee beans, which is dependent on many factors, some of them intrinsic to the plant and growing conditions, but other dependent on the post-harvesting processes and also brewing and serving methods. So in terms of processing, that is a topic for today, post-harvesting is a critical step that significantly improves the bean quality, including all flavor and aroma. And among these stages, fermentation plays a crucial role because the microorganisms involved in this process are responsible for produce most of the flavor, but also the flavor precursors in the coffee beans. And taking the example from the wine and from the beer industries, many coffee producers has begun to use startle culture during coffee fermentation to ensure the best quality, but also to modify or generate special characteristics in coffee products using innovative process. Other important thing to take in mind in coffee is that traditionally coffee has been consumed as a hot beverage. And this is the common way that we consume coffee, in my case, and in Colombia, I think. But coffee, in this case, coffee market is growing in new categories. It's changing from hot to cold brews products and also ready-to-drink products. And this is mainly to attract millennials and centennials, offering new ways of consumption. So this means that recent trends include not only the coffee beans, but also the development of ready-to-drink beverages using coffee as a primary ingredient. So it's common to see the concentrated liquid coffees, and these products are quite trending in countries like Korea, Japan, USA, and have different challenges about the stability, sensory profiles, and differentiation or provide different alternatives for the consumers. So today, as I mentioned, the market of coffee is not only working as a as a traditional hot beverage. We need to think coffee in a different spectrum. So we have a different market spectrum from traditional hot brews to instant and soluble and also ready to drink products. And also people is thinking in coffee not only right now for the sensory profile, but also for the functionality that could have for our health. So we need to think in these different options. So in this case, each product requires consistency, differentiation, and also a deep understanding of how processing affects the chemical composition of the coffee. So this is where metabolomics become especially valuable because allow us to move from a general quality descriptor of to a more precise molecular understanding of the coffee beans and also of the transformation during the industrial processing. So I'm going to show you some examples of how we approach the improvement of these processing technologies using untargeted metabolomics approach. So let's start with the traditional coffees. And in this area, the first application I would like to show you focus on the specialty coffee fermentation, where we evaluate a novel fermentation strategy known as culturing. And in this study, we aim to assess the effect of inoculation, supplementation during coffee fermentation on the coffee quality, the microbial communities, and also the metabolic profile of the coffee bean. So to do this, coffee bean fermentation were performed in a farm located in Risaralda, Colombia, owned by UVA company. This company produced the traditional wash coffee as well as culturing coffee. So culturing is a fermentation method developed by the producer, which involve the use of starter culture, but also they use fruit as an energy substrate for the microorganisms during the fermentation, and this process occurs in a tanks with using pulp coffee. So here is an overview of the experimental setup. So we conduct the experiment in the same protocol that the company do and we compare the standard fermentation protocol and the fermentation using the starter culture and also the inoculation with the fruit as a substrate. We're collecting samples from the beginning and the middle and in the end of the fermentation process for DNA extraction and also metagenomic analysis. And then we collect the beans and sun dry the beans during 15 days, collect some beans, and then these beans were roasted. And these two samples were employed for untargeted metabolomics. And at the end, we prepare the coffee beverage and perform the coffee cupping for the sensorial analysis. So the DNA extraction was performed by the Computational Biology and Microbial Ecology Group at UNIRIO and also the DNA Innovation Laboratory at Washington University. This employs a standard protocol for this type of analysis with a power soil isolation kit from Qiagen for DNA extraction. And then we perform metabar coding for before and ITS1 regions, and the sequencing was carried out using a platform, Illumina MySeq platform, and all the bioinformatic analysis was performed using SHAME2. And the targeted metabolomic analysis was performed in our facility. We employ a standard protocol for coffee analysis, coffee extraction, and the conventional analytical plan for using RiverFX and also Helix analysis in LCMS and also GCMS and the conventional untargeted metabolomics workflow for data processing and data identification. And you're going to see that across all the examples that I'm going to present, we always in coffee use the sensory analysis that is really important, and also other parameters that the industry use. So in this case, coffee was done by certified Q graders, following the standard protocol established by the Specialty Coffee Association. So what we observed in this study, so we found that the coffee beverage for the standard process were more chocolate-like in flavor. And also we observed that this coffee is less sweet, more acidic, and had some note of caramel, coconut, and almond notes. But here the panelists detect an important thing that was a defect corresponding to a phenolic known. In contrast, with the process, with the coffee obtained from the inoculation, we observe a more floral flavor and in aroma for this coffee. We observe a increase in sweetness, a more clean cup of coffee, and also with a best uniformity. And the important thing that was a cup of coffee free of stains and defects. So from the sensorial part, we found that the inoculated fermentation resulted in a better cup of coffee. So now we want to look in terms of metabolites, what means that. So then targeted metabolomic analysis show a clear separation between the standard fermentation and the inoculated process. We identified 59 metabolites that were significant in this process, corresponding to amino acids, aromatic, carboxylic acid, cinnamic, and canic acid, among other families. And here we can observe the correlation between many of the differential metabolites associated with a specific organoleptic profile found in coffee beverage. For example, we observe higher abundance of metabolites associated with sweetness in the inoculated coffee, such as fructose, sucrose, or vanillin. In contrast, we observe higher abundance of metabolites associated with this caramelic fruit, bitter, and acidic note. And moreover, the standard fermentation show a higher abundance of metabolites related to bacterial metabolites, and the inoculated process an increased composition of metabolites associated to fungi metabolites. We conduct the same for the roasted coffee. We here identify 77 metabolites, and here in the same approach, we do a correlation between the metabolite, the composition to the attributes in the coffee profile. And this is important to understand the coffee and to explore how each type of microorganism develop different type of metabolite, how this impact the sensory profile. So the last step was to explore the microorganisms involved in this process. And in terms of bacterial community, we observed that mainly at the beginning of the fermentation was mainly composed by lactobacillus and leuconastoph. And in terms of fungal communities, we observed Saccharomyces staphylococcus and also Castanea. But during the second phase of the fermentation, we observed that an increase in lactobacillus in both processes. But in the case of inoculation, we observed that a higher changes that Castanea was the most dominant microorganism during this fermentation. And at the end of the fermentation, we observed no significant variation in the bacterial diversity among the process, but we observed a significant variation. And also we observed that Castanea humilis emerged as the dominant yeast in this process. So in this study, we demonstrate that the inoculate fermentation resulted in a better cup of coffee, free of stains and defects. This inoculated fermentation process improved the metabolite composition associated with desirable notes in coffee. We know already what are the microorganisms involved in this process, and also the relationship between these microorganisms and the metabolites produced for the coffee. And we are working in the complete understanding of the influence of the microbiota and the coffee quality. So we are working or exploring the interaction between the microorganisms, the isolation of possible strains that complement the current commercially option for starter cultures, and also to produce new and innovative flavor profiles. So here is really important to know what happened with different types of microorganisms because we can work in product designs depending on the consumer demand. So together with Cold Cafe, which is a company that uses green coffee. I don't know if it's common for you what is green coffee, but if not, green coffee means is the coffee that already has been dried but not been roasted. So with Cold Cafe, we want to make a first exploration to determine if it's possible to modify the metabolite composition and also the flavor profile in the coffee to get some or develop some specialty like coffee profiles from regular or from commercial coffee. So this work was entitled Enhancement of the Fruity Flavor in Coffee Beverage by Solid State Fermentation in Green Coffee, multi-block approach. So to do this, we perform an analysis using 3 blocks: the physicochemical block, the analysis of volatile compounds, and also the non-volatile compounds in this case. And we want to explore what is the effect of this fermentation. So here we can observe the experimental design for the evaluation of solid state fermentation of green coffee. We use raw green Arabica coffee in these experiments. We use also a commercial yeast, a Saccharomyces, and this yeast was hydrated with water before the experiment and incubated at room temperature during 15 minutes. And then it's an important step that the coffee were first steamed during 10 to 30 minutes to achieve a moisture that is at least 25% in the coffee beans. And for the fermentation trials, we select conditions based on a screening experiment that we generate different levels of fruity flavors. So then the coffee was dry and stored, and at the end we had 12 total samples from fermentation process. Here we have some controls or unfermented coffee and other controls that were named as a reference because corresponding to 2 commercially fruity coffees that we have in Colombia. And for our fermentation, we perform biological replicates. So here is an overview of the multi-block analysis. I'm going to explain each one, but I want to show maybe this is quite not common, that this is the analysis of volatile compounds coupled to olfactometry. So here the volatile compounds were extracted by solid phase microextraction, and then for the olfactometry analysis involved 5 panelists that were training in these coffee aromas with a kit which include 32 essential coffee descriptors. And these panelists evaluate the aromas detected at the ODP port, and they were required to recognize the aroma and also to provide a descriptor recording the retention time and also intensity on a scale from 0 to 3, where 0 is not detected, 1 is weak or barely recognized the other, 2 is clear but not intense, and 3 is intense. So let's see the results. Here we observe all the physicochemical composition in the green, roasted, and also in the coffee beverage, and we saw significant changes between the fermented samples and then fermented samples, and also to the reference of control samples. We observed these changes in terms of acidity. We observed that the fermented samples show lower pH and higher T-table acidity, and this effect was strongly in the high fruity category. We also found that water activity remains below 0.7. That is important for all the microbiological stability. In terms of sugar, we observed that sucrose decreased. That is expected output in this process, but also we observe an increase in reducing sugars like glucose, fructose, and this is important because show us the active metabolism of the yeast. Caffeine is another parameter that is important, or a routine measurement in this industry. So in terms of caffeine, the content of caffeine remains stable across all groups. And finally, in terms of chlorogenic acids, which is another quality control parameter for coffee in industry, we observed that fermenting categories show higher chlorogenic acid contents. And this increase was most evident in the highly fruity samples. So then we were to non-volatile compounds. We identified a total of 77 important or differential metabolites in these experiments. Here we can observe the result from the olfactogram. So here we can observe the 47 compounds that we identify in this analysis that as having an aroma activity, and each chromatographic bit corresponded with this with the corresponding aroma. And among these 47 compounds, we identified 7 that are really important as aroma descriptor for floral, fruity, caramel, nutty, but the most important one for us, the fruity flavor. So in terms of the sensory analysis, we observe an important result, that is that for this blue line correspond to the high fruity samples obtained from the fermentation process. So we observe a significant increase from around 2 to around 6 score for fruity attributes compared to the control samples and also with the reference samples. So this is quite important. And also we observe that this increase in this attribute have an impact in the overall score for coffee. So we confirm that fruity attribute is highly desirable in coffee, and a commodity coffee quality can be enhanced to deliver novel profile. So this was an important result for this study. So finally, we want to integrate all the parameters that we measure and try to develop a model to establish the impact of all these variables in the development of the fruity nose. So here we construct a model using 103 features corresponding to 12 physicochemical variables, 14 volatile odors, and 77 non-volatile compounds. And here is an easy way to present the summary of this model. So we found that the key markers associated with fruity attribute in terms of physicochemical markers correspond to treatable acidity, fructose content, and the content of some key chlorogenic acids. Also, we identified the most important volatile markers and also the most important non-volatile markers corresponding to carbohydrates and also kinic acid derivative. And here we can observe the contribution of the data of each block to the model that help us to understand more the fruity attribute. So in this study, solid state fermentation enabled the modulation of the fruity flavor in green coffee and also the overall score for this coffee. So fruity intensity score increased significantly in this process. We observe difference in terms of fructose, kinic acid, pH that are quite important in this process. We observe also another difference that is important, is a decrease in flavonoids and organic acid and derivative in fruity samples. And we have a better score in fermented samples. So the important thing in this study is that we provide or prove that this solid state fermentation is possible to modulate fruity flavor in green coffee, and for us open the door to explore other modulation in other attributes. And we already know that is possible to increase the quality in these coffee beans when we are talking about green coffee, not only in fresh coffee. So now I want to move on the applications of metabolomics in these ready-to-drink products. In this case, I want to show two examples. One, when we work on shelf life, in this case, we explore metabolite markers associated to some rejection in some coffee products. And another example that is evaluating new technologies to generate different products or new ingredients for ready-to-drink products. These both examples were also in collaboration with Cold Cafe Company. So the first one is to explore the metabolites or the chemical markers related to the acceptance or rejection of coffee beverage in these concentrated liquid coffees after storage. So here we first conduct the sensory analysis and we divide the coffee samples from the accepted and also the rejected samples. And we perform the conventional analysis in these coffee studies and targeted metabolomics, but also all the physicochemical characterization that are common in this industry. And we explore what happened. So here is an overview of how these concentrated liquids are prepared. The sample were prepared in a coffee roasted in a food processing plant in Cold Cafe, Colombia. For this process, we use a percolation battery that involves 1,800 kilograms of coffee per extraction. This is a reason why we use as less as possible replicates in this industry application, because we are talking a lot with high amount of samples. So then we obtain the coffee extract here. We pasteurize these coffee products to avoid some microbiological spoilage, and samples then were stored during 120 days. Some of them at room temperature or 25 degrees Celsius, and another at minus 30 degrees Celsius. So again, we performed all the physicochemical properties and also the sensory analysis to classify as accepted or rejected samples. In the same way, we performed the targeted analysis using a multi-platform approach, and we observed that 80 metabolites were differential between the both products, and we observed a clear separation among them. And then we start looking about the mechanisms involved in this product degradation. So we also integrate all the parameters. We found the attributes related with the acceptance, but more important with the rejection of this type of coffee samples. And here we found a lot of compounds that are really important in the coffee quality. So with all this information, we can map all the degradation reaction in this process that were related to the degradation of chlorogenic acids. This degradation promotes spoilage in this type of sample. And this happened because when chlorogenic acids has hydrolysis, they produce or release ferulic acid, quinic acid, and some camphoic acid. And these compounds trigger different degradation pathways. Some of these pathways lead to the formation of some compounds that are methoxybenzaldehydes and also hydroxyquinone and other related products that are undesirable in coffee products because produce or affect negative the sensory profile in this type of products. So with all this information, these results support the product development and innovation for the company, helping them to implement new strategies to understand what happened during the process, and also to develop new ways to to improve the shelf life, for example, using natural ingredients that prevent the degradation of this chlorogenic acid. So as a result, at the end, the company was able to increase the food product shelf life in this product for several months. So this is really important in this type of industry. And the other type or other example that I want to show you is to how we use this untargeted analysis to explore the application of new technology in this type of products. So this performed this study entitled Uncovering Metabolic and Sensory Changes in Coffee Extracts via Ultrafiltration Membrane. This was a study because the company wanted to explore other technologies to produce different type of ingredients for ready-to-drink products. So consumers want different type of coffee products. Some prefer more acidic, other more floral in flavor, other more chocolate, other more strong. So we need to develop different profile. So they want to use this technology that is quite common in food industry, but in milk concentration or juice clarification, and also in seawater desalination, but it's not exploring coffee products. So we performed this study to evaluate if ultrafiltration membrane produce a different profiles in these coffee permeates using this type of membrane. So here we use, this is wrong, but here we use the different type of coffee extracts that the company have. For example, the cold brews that are roasted coffee extracted at low temperature. We have also the freeze concentrate, that is a coffee that is extracted with water close to 140 degrees Celsius and concentrate at cryogenic conditions. Then we have the evaporated extract, that is a coffee extract with water above 150 degrees Celsius, and then is concentrated at temperature close to 50 degrees Celsius. And we have the lightly roasted evaporated, that is a coffee extract above 150 degrees Celsius, and also evaporated using spray dry and light roasting. I want to show you this because I want you to see the different options that we have in the industry to develop different profile of different products. So we want to evaluate for each one of these coffee concentrates what happens when we apply ultrafiltration membrane. So we use and pilot scale processing plant for this ultrafiltration membrane. We use 60 liters per experiment. So we start the processing, and at the end, using these membranes, and at the end, we had 2 samples per experiment, the feed, and also the permeate sample, that is the sample that is important for us. So we measured, again, physicochemical properties and also the sensory analysis. Here we developed a most robust sensory analysis with around 52 attributes, because it was really important for us to perform a correlation between all the metabolite composition and the different profiles in coffee products. And for them, for the industry, it's possible to develop in some machine learning tools to predict different profile if they use different industry or operational parameters. So here again, we performed a targeted analysis, and I want to show you here the sensory results. I want to present only the 11 most significant sensory attributes because it's easy to present. And the blue line corresponds to the feed sample, and the red line corresponds to the permeate. This is the most important for us. So overall, it was found that the treatment with this membrane resulted in better or positive changes in the sensory profile. Here we can observe easy. Okay, that for all type of coffee extract, we observe a reduction in these undesirable notes like paper or also vegetative or also tobacco, and an increase in general in notes that are desirable, for example, floral, fruity, in some cases in acidity, caramel, and in general in the global score. For example, for this evaporated product, it was a really nice result in which the permeate increase the overall score for these coffee products. So what we see in terms of the physicochemical changes, we observe a reduction in the TDS sample. This is important because this leads not only a reduction in this, but also in the turbidity. And this is important because this technology clarifies the coffee extracts, and the appearance is important for the consumer. And also an important parameter is that we obtain high caffeine content. So the main challenge in this type of product that consumer wants high content in caffeine, but a nice sensory profile in the product. So here we get this option or higher caffeine content. So again, we do all the untargeted approach, analyze all the metabolic changes in this process, and also integrate all the information to explore what happened. And one thing that was surprised me is that the first component that separate these groups were the roasting degrees of each type of coffee extracts, not ultrafiltration membrane. So it's most important, this effect. So here we analyze what is impacting the roasting degrees, and this was another information for the company that we don't expect to get. And in the second component, we observe the effect of the ultrafiltration membrane. So we observe in this part all the feed currents, and here all the permeate currents, so we can understand what are the attributes that are involved in this process. And also we observe a component 3 that is more related at the temperature of the extraction. So here they observe also different parameters that are quite important or modify the metabolic profile in not only in the ultrafiltration membrane, but also during the coffee extract production. So one important thing is that in this case, the permeate current resulted in a really nice product that improves acidity, bitterness, and also enhance attributes that are desirable in this type of beverage, like nutty, fruity notes. We observe a reduction in total dissolved solids. That means a reduction in turbidity in these products, along with an increase in caffeine concentration. And this tool, or this untargeted approach, offer a valuable insight in how is the correlation between the metabolite composition and the sensory profile using this technology. And one of the important things is that despite all this alteration that we observe, all the permeates maintain their identity as a coffee product. So it's possible to use a coffee ingredients in this type of ready-to-drink beverage. So at the end of these studies with this company, we observed the application of untargeted metabolomics in different points, from a profound sensory understanding of this matrix composition, but also to work more in know the product, and for them it's possible to design products before the experiment. So metabolomics allows a product description that reveals the potential of these products, and also to explore other types of technologies that were really critical. And it's important to understand all these capabilities for this abundance before implementing in the company. So for Cold Cafe, with this research, it was possible to go from research, from basic research, and also using omics science to go to the product development. So for us, it was an amazing collaboration. And over this year, we have learned that we have different options in coffee products, but without any doubt, for me, fermentation is one of the most powerful strategies to transform coffee, not only in terms of the sensory profile, but also in this metabolite composition that can be or can have a bioactive compound or maybe functional properties. So right now, the market is not longer seeing the coffee as a beverage that with a good profile. It's also important to think in the functionality, the effects that coffee has on our health. So with this in mind, I want to go to the final part of my presentation, that is to talk a little bit about the next step for us in coffee research. And this step is part of the Juan David Derek's doctoral thesis. So we want to use omics approach to understand fermentation role in enhancing not only the sensory attributes, but also the bioactive and functional properties in coffee using different types of fermentation. So the first step to do that was to conduct a very detailed review of everything that has been reported for coffee in terms of bioactive and functional properties. That is new for us. We had a lot of experience in sensory attributes, but this bioactive is and functionality is quite new for our research, and also it's a new topic. So the aim of this review was to explore how both the spontaneous fermentation, the conventional ones, and the novel fermentation have an effect in these coffee products in both aspects, sensorial and also functionality properties. So here is the methodology of the review. This review is under revision, so I hope that is available soon. So here we at the end get 25 papers that were talking about coffee, fermentation, and only, or the focus were functional and bioactive metabolites across all the different types of fermentation process, the conventional ones, natural processing, honey, wet, solid state fermentation, but also the special fermentation process. So I don't want to go in details in this review. What I want to show you is this final input that is really important to us, and is that in this figure we observe the summarize in terms of metabolites that are measured to evaluate bioactive properties and also functionality across the different fermentation types. And we observed the type of biological activity that were evaluated. So what we observed in this review is that commonly research in this type of approach always measure the common metabolites, caffeine, trigonelline, and all the chlorogenic acids, some phenolic acid in targeted approaches. So there is a lot of information missing in terms of metabolite composition that can be bioactive or have functional properties. So that is the reason why we want to apply a targeted approach to explore other metabolites that could be really important for this type of application across the different fermentation types. And here, for example, we saw an overview of the biological activities and the most studies antioxidant activities with total phenolic compounds and the common measure for this type of application in food. But there is an increased reports exploring other biological activities than neuroprotective or anti-inflammatory, but we have a lot to do in coffee research thinking in these topics. So with that in mind, we get our list of the important metabolites that can be reported as a bioactive, and we want to explore the other information. And we decide to go first in all the type of fermentations. We went first with this type of fermentation that is post-drying fermentation. I think that this type of technology is a new opportunity for innovation in coffee processing, because traditionally we ferment, or the fermentation is close linked to the harvest. So normally this takes place with the fresh fruit, and this has some limitation. What we want to continue exploring is to ferment the sample after this step, and also after storage in dry coffee beans, because this presents some advantages to or flexibility to improve the quality of the beans. And these have a lot of opportunities, not only in some type of coffee, also in the different coffee varieties. For example, coffee Arabica is a well-established coffee variety with well-known sensory profile, but we also have coffee Arabica with low quality. But for example, we have coffee Canephora, that is a coffee variety that is, and it's really important in terms of production, especially in Brazil, for example, and have a really good climate adaptability. But the sensory profile is not that good as coffee Arabica. So we have different options using this post-drying fermentation, and also because using this fermentation after the drying process, we can also innovate or modify a compound beyond the harvest season. Also, we can create on demand different micronodes using different microorganisms. And we work in an important thing that is lot revalorization. So we don't have a lot of coffee. So we need to start thinking how to improve the sensory profile on this type of varieties, for example. So with this in mind, we start the first part of the Juan David doctoral thesis, conducting this study that look for improve the flavor, but also the bioactive profile in low quality Arabica and also in low quality Canephora. This is the common workflow for solid state fermentation. It's the same that I presented in the previously study. So here we are trying or testing these two low quality coffees, and we are using one type of yeast, one type of bacteria, and one type of mixture of the yeast and bacteria to explore what happened in the beans. And we analyze the same green coffee and also the roasting coffee. So for this type of analysis, we are using our new capabilities with this tribrid system that we are going to focus on. The first one approach is to improve the metabolite annotation capabilities, and then we want to use the SQUAD analysis, that is a simultaneous quantification and discovery analysis, to work in the exploring new metabolites in this process, but also to focus on this metabolite that we collect in the previous review work that are important to bioactive compounds in coffee. So for untargeted metabolomics, in terms to increase metabolite annotation capabilities, we are running all the new libraries for these 1,500 metabolites that are most common in secondary metabolisms. And then also we are using the MSN tribrid capabilities to identify the unknown compounds. And also we are looking for all the bioactive compounds. We are working using this SQUAD analysis. I don't know if some of you are are related to this type of application. If not, SWOT analysis is a relative new workflow which combine both and targeted workflow. This is as a technique in which we define a predefined set of metabolites for targeted metabolomics. In our case, this set corresponds to the metabolites with by activities that Juan David collect in the review work. And then we also conduct the untargeted approach for discovery. All this using a single injection. So we use this tribrid capability in the orbit trap, the untargeted analysis, and in the linear ion trap, the targeted quantification. And for targeted quantification, we are analyzing these metabolites in coffee sample. So here we have some previous results from coffee Arabica. We see the first important thing that is a difference between fermentation process and the unfermented sample. And here we can observe that we obtain different coffee profile from the same bean when we use different microorganisms, and we are exploring what metabolites. But for Robusta, we observe an important thing that is a difference between the fermented sample versus the unfermented samples. But here we observe that the metabolic profile is almost the same using across the different microorganisms. It's almost the same if we use a bacteria, yeast, or the combination of both. So we are looking in this theme. But I don't want to spoil more results today because Juan David will be in Buenos Aires presenting these preliminary results. So I invite you to visit the poster in this Metabolomics conference and to learn more about this ongoing research. And I think we are on time, so I want to close this seminar and I would like to summarize with some take-home message about this application of untargeted approach in coffee research. So I think the key message is that metabolomics help us to connect process chemistry with all the sensory perception and product innovation. So it allows us to understand how process like filtration, fermentation, new technologies in industry, storage, transform the chemical profile in these foods, in this case, in this coffee, and how this transformation affects the aroma, the acidity, and or the consumer perception. So I think it's important to remember that metabolite display not only what is present in a product, it's not only for metabolite characterization, but also it's really useful to understand how industrial process transform food chemistry and how we can shape these products for a better consumer perception. And I want to acknowledge to all our collaborators in coffee research. This is the team from Cold Cafe. This is our team Nuniandes. Right now, Professor Andres Gonzalez is directing with me the Juan David's doctoral thesis. Here is the MetCore group. So I really want to thanks to all members of MetCore, especially to Juan David and Ana Maria Leon, that are the PhD students working in this type of application. And of course, I want to thank you all for your attention.
Monica, thank you so much for your talk. It's a really impressive work since the samples to the data processing, the results, and now the target metabolomics. I'm shocked that you can identify as level 1 1,500 compounds. So it's a huge number. It's so impressive. Please, to the audience, if you have a question, the QA box is open to make question. We already have some of them. Myself, have a lot of questions, but we can talk later. So the first question is, which software tools are used for this work analysis?
For metabolomics analysis, we use the conventional ones. It's depend of the machine. We use MS Dial as common tool for this data processing. And then for all the statistic analysis, we use some package in Python and also using R Studio. So we have this software. And right now for squat analysis, we use the software from Thermo that is compound discovery to get these two options for untargeted discovery and also quantification. It's depend of the data. And of course, we are building this library, so we use our PCDL software for our in-house libraries.
Amazing talk. So I have a question before the next question. So um, as a growing researcher, one of the questions that I ask myself is, how do you link? How happens the link between the industrial uh, and the researcher? Do you seek them out, or the industry um, talks to you, or they have a problem and they're searching for someone to solve it?
Now, I have to confess that at the beginning of MetCore, the university wants to work closely with industry. So this is not common in Colombia. We want to show the industry that research is important and we need to innovate in our products to give more value. So this was a task for me at the beginning. And for me, it was like, it's going to be impossible because I think it's difficult. And with omics, for me, it was like, I don't know how, but I think it was really lucky that I met some people from this industry and also from cocoa industry. And this industry start some research group and innovation and get for them their research group some PhD. Um, so they researchers were interested in know this capability. So we conduct one study. At the beginning, they only, for example, in this case, want to know what happened with this degradation. This was the shell life was the first approach. And when we saw that with a small, well-designed experiment, we were able to know what happened in a process and to solve the problem to increase the share life in a product is a lot of money for the industry. They know that it's really important to know what happened in this thing. So for coffee was like with these first services that they contract us at the beginning, they were in love with the metabolomics capability and they want to start using in this part. And for me, it's an amazing thing because it was almost the same for cocoa thing because some industry are thinking understand profoundly this matrix and also, as I mentioned, developing machine learning algorithm. And for example, if a consumer say that I want a coffee with this profile, maybe if I know which microorganisms produce which profile or which process, I can go back and develop a product as the consumer demands. So I think it was easy part. It was a complement between MetCore looking for the industry, but also there is interest in industry in improve their process.
Thanks a lot for the answer. So the next question is, will the slides be available? On, I don't know in your case, but the recording will be available on the YouTube channel.
Yeah.
So if you want to go there, you're welcome. So the next question is, why are you not complement your metabolomics with NMR metabolomics?
This is a common question, but the first true answer is that it's because we don't have NMR capabilities, but the second one and the most important is because in this application, when we work to mapping in detail all the mechanisms, all the pathways, all the reaction involved, I think that mass spec have more capabilities to go in this detail. We use MMR, but mainly in, for example, this application for origin denomination in this application when we need more and more robust and reproducible tools. So we use NMR in this type of application in foods. When we want to build a model to predict something, we need a robust technique as NMR. And for this mapping, I think MS give us more capabilities to mapping a lot of compounds, for example, in lipids. I don't mention a lot in lipids, but lipids are really important in cocoa samples, so we need to go to mass spec. That is the reason.
So another question is, how is combined LC-MS and GC-MS DTA?
So I think it's data, but we combine this at the end. We didn't. Okay, this depends. If when we work with using GMPS or molecular networking, now we can combine the data, but we analyze the data of each platform and they combine all the important features, because if not, we are going to put a lot of features in statistical models, and this is a bigger problem for overfitting, for potential overfitting. So we first select the important ones, metabolites in each platform, and then going with these important ones to combine all the results, also with the parameters that are common measuring industry. And this is an important thing. We can do these omic studies, but we always need to measure some parameters that industry can monitor across the process and is common for them, because in this way we can connect. Maybe it's not because it's not possible using in quality control metabolomics or untargeted approaches. It's so expensive. So we always try to connect the data with some typical parameters in this industry process. So we connect the information is during the statistical analysis, not before.
So the next question is, there any specific phenolic compound that is more related to the roasting level of the coffee bean?
But I don't remember right now. Maybe Juan David's here in the chat, but I know that we have the roasting level. The effect of roasting level we saw more related with volatile compounds. So this is the main point. So, for example, if you remember the graphic in membranes, the roasting temperature were more associated to volatile compounds, and maybe some compounds that give this strong notes, and for me are not really good, so I prefer lightly coffee, but it's more related to volatile compounds more than phenolic compounds.
The next question. First, congratulations for the nice talk, Mónica. Just for curiosity, if you could choose just one or two metabolomic marker for routine cough quality control, which ones would you select and why?
Thank you, Alexandra, but this is a difficult question because it depends on the process. I think that the quite common are the chlorogenic acids, but we really want to change that. We really know that are many more metabolites that are important, and thinking, for example, in these small peptides that are really bioactive. that are important also in fermentation. So it's depend, but always chlorogenic acids, always some phenolic compounds are important in this flavor profile. So it's depend. It's depend of the process that we need to measure or we need to control.
So next question, did you work in any project related to coffee authenticity? If so, what are the adulterant biomarkers you believe will be interesting to investigate?
And not yet. We are working in authenticity project, but in our agrobiodiversity products, we are working more on a type of onions that we have in Colombia. This is an unique type of onions in our country, so we are working on that. But we really want to do that. I was a few weeks in Brazil in an event in Minas Gerais for specialty coffees. So I was really not surprised, but I was really happy for me to see all the improvement in this coffee research. So we really want to start working, analyzing the different varieties of coffee, looking for coffee authenticity, because it's a lot of work to develop this type of products. Also, I know that there have been studies to differentiate coffee for different countries, but right now this is more difficult because it's not only a type of coffee from Colombia. In Colombia, we have, I don't know, I think 20, 40 different types of coffees that we produce. I think it's more, but it's the same in different countries. So this is a point that I really want to do, and we are trying to connect around Latin America. For example, last year we were in a school in Cartagena in food metabolomics, and we met people working in coffee from Mexico, from Guatemala, from Costa Rica, from Brazil. So that was an idea after this meeting. So we are trying to establish some consortium around coffee metabolomics to start doing this type of analysis.
Before one more question, nice comments. This is not a question, but a compliment for your important work and research and for everything you do and contribute to science. And now the question, do you have trained panelists in your lab or you get the panelists from outside?
Both. We always work with the panelists that the company have, because some companies have a specific panelist for their specific products. So it's complete different testing the traditional coffee or specialty coffees, or testing the cold brews or ready to drink products. For example, Concafé have their own panelists, but we have our sniffer. The picture that you saw in this olfactogram is Juan David, actually. So right now Juan David is working with us, and he's the expert in coffee research. So we have some people that knows, but we always go to curators, or maybe send to other, for example, in membranes, we need more attributes, so we need to send a company that is specialized in this type of coffee. So I think it's the important thing is to have people that is trained and always using the international protocol for this type of coffee.
So first of all, congratulations on the great presentation. I was wondering whether the new biomarkers identified in your studies are eventually implemented by companies as quality control markers in routine analysis.
Yeah, that's the point. That's the point. And for example, in terms of this type of shelf life study, we have some specific compounds that are implemented in the routine using common analytical techniques, LC coupled to ultraviolet or DAD, for example. So we need to translate to these applications. I don't show here, but for example, companies in coffee use NIRS a lot to differentiate the authenticity, to differentiate Arabica versus Robusta, and there are some markets that are implemented. But for this study, the main goal was to understand what happened and explore new ingredients. So right now, the membranes from the other studies, we don't have biomarkers or markers in quality routine because this was not the aim. But this is the idea, try to establish some important metabolites and go to conventional analytical techniques, and in that way it's possible to use for the industry.
One more question. Did you do metabolomics in the coffee waste to transform it?
Yes, actually, this is the part of the thesis of Anna. Anna did all this coffee research as her master thesis, and right now Anna is working in strategies using omics tools to improve some waste products, mainly from coffee, of course, and also for cocoa. So yes, we are trying to develop some workflows to valorize these products using untargeted approaches, thinking in functionality and thinking in functional products.
So another question, how could metabolomics studies in coffee contribute to the development of soil management and conservation strategies in coffee growing systems?
I think there is a lot of option and studies in that, because we are actually, Brazil is working a lot in this option to trying different type of varieties that are in crops with different ambient conditions. So I think metabolomics is the perfect tool to understand all the environmental factors, not only for us, also for plants. So we can measure all the abiotic, ambiotic parameters for plants. So we can measure and testing, I don't know, the less water in these crops and different heat temperature conditions in these crops. So I think metabolomics is the perfect tool to explore this option for conservation strategies.
Another question. Thank you for your talk. The focus of the presentation was on post-harvest techniques and influence. In your experience, how much does the type of coffee bean chosen to work influence the post-harvesting results?
I think completely, always. I think this is the main point. We had, I think one of the most important thing is the type of coffee. For example, as I mentioned with Arabica and Canephora, we can have a low quality Arabica, but normally, because I taste one of the best Canephora coffee in Brazil 2 years ago. So normally Arabica had a really good profile, even if it's a low quality Arabica, in fruiting some attributes that are common to the genetic to the plant. And for example, Canephora is a so important crop, but the flavor profile is not that good. So it's completely different. Right now, for that reason, we are measuring also Arabicas and Canephoras with the same conditions, because we want to start understanding that, because as you saw, we see with the same microorganisms, we see a complete preliminary output of the fermentation. We saw that Arabica produce different profile with this microorganism, and for Robusta, not. So why, we don't know already. This is the task, but we know that that first important thing is the bean. But then the nice thing is that we don't have a lot of coffee around the world. So when with all the climate change, we need to work in improve the coffee that is not good as canephora. So the important thing is that with fermentation, we are capable to modify this profile, and we know this right now. So I think this is open the door to explore different consortium. We also working with a professor from Brazil in another doctoral thesis focused only in canephora, exploring at least 20 microorganisms and know what happened, understanding all the process. So we can modify in different steps, but for sure the bean, the type of variety, it's one of the most important parameters.
Okay, to finish, I have one question. You said that now you were trying to integrate on-target metabolomics and target one. I want to know about you. I know you have a huge number of standards to do the quantification, but what was the major challenge in this transition, in this integration between the two types of metabolomics?
I think all because it's a new approach that we have. So all is a challenge from the start working with this tribred machine that is an amazing for me. I have to mention that I really love this instrument and this capability. And for me, I didn't know before the instrument arrived, MetCore, that is an option to this instrument. And when we saw this option, for me was so amazing, not only for coffee, because in foods, we always want to do untargeted, but also looking for some important metabolites. So I think that also for the industry, this opened the door to put in one solution on things. So I think it's a challenge is to have the standards, but we are in this process of training because the machine just arrived in December last year. So all the data is really new. So Juan David is working a lot to have all the data, but it's not so difficult because it's the same thing that if you have worked in targeted analysis using, for example, triple quad experiments, it's the same. You need first get you the standard and running the samples, and you have the retention time and the precursor, and then you can use the conventional target parameters and put the exclusion, the targeting list, so the machine conduct both in parallel. The machine is measuring the orbit trap, and in parallel is targeting this analysis in the linear trap. So the challenge is to get the standards, and also here you need to separate the targeted component that you want, because you're going to measure in a low resolution part of the machine, that is the linear trap. And then you can only measure the points in calibration curve and conduct both together. So it's really, I think using the software Calmo is quite simple to do that.
So, Juan, do you have any questions?
I don't have any more questions, but I want to thank for the most amazing talk. So let me share my screen for the last slide.
So on July 31st, we'll have the next webinar, the July webinar after Metabolomics. So please, if you want to participate, the link registration is here, or you can click in link by LinkedIn or other social medias.
So Dr. Justin Carrer will be joining us in his talk is about metabolomics and lipidomics in sport and exercise medicine. He's a senior physician in sport and exercise medicine at the Lausanne University Hospital, also head of the sport and youth health unit within the Swiss Olympic Medal Center. If you want to join us, just go to all social media and the link is over there. So thank everyone. Have a good day.
Thank you. I hope to see you all in Buenos Aires.
I'll see
Bye.
you there. Bye.
Bye.